All right. Thank you, everyone, for joining us today for the first OHBM webinar of 2024, where we'll be looking at the current funding landscape for neuroimaging research in Australia. To sort of get us started, I would like to pay our acknowledgements to our country. So in the spirit of reconciliation, we would like to acknowledge the traditional custodians of our country throughout Australia and their connections to the land, sea, and community, and pay our respects to their elders past, present, and future. And as many of us are across different lands in Australia, I'd like to acknowledge all mobs and communities across Australia and extend their respects to all our Aboriginal and Torres Strait Islander people today. So as I just mentioned before, I'm Natasha. I'm a postdoc at University of Sydney with the Brain and Mind Centre, and I've got a little poll there asking you to fill out what your research level is, and we've also dropped in the chat a survey questionnaire on what future research topics you would like us to cover in the OHBM webinar series. So I'd greatly appreciate it if you could fill that out. And we're going to get started today on introducing some of the panelists to this discussion. So today we've got Dr. James Pang from Monash University, Dr. Cassandra Wannan at University of Melbourne, Dr. Remika Mito at University of Melbourne, Associate Professor Jess Taubert at University of Queensland, and Associate Professor Max Shine at University of Sydney. And I would like to firstly introduce you to James, and he's going to give us a sort of brief overview of his research journey thus far.
Do you want me to start sharing now?
Yes, if you're able to.
Right. All right. Shall I just go?
Yes, go ahead, James. Take the lead.
All right. Ooh, it's pretty difficult to start this one. First, thanks again to OHBM and Australia for inviting me here. I have the imposter syndrome at the moment, because I feel like I don't know why I'm part of the panelists, but anyway, I think I'm just gonna share a little bit of what I've experienced and a little bit of what I have done to give you a little bit of context of the successes and the failures that I've had over the last couple of years with grant applications. So I work at Monash University with Alex Fernita, and really focusing a lot on neuroimaging and computational neuroscience. That's pretty much the combo that I always do. So during my PhD, I was really focused on, computational modeling of more kind of microvasculature. So doing a lot of hemodynamic modeling based on fMRI and stuff. So the reason why I'm sharing this is the things that say the papers, for example, that came out of this PhD, in my opinion, was cool, but obviously it's not as impactful as you would have thought, maybe if you want to apply for something more health or medical related or something. So I had a decent number of publications after the PhD, which is probably what kind of led me to some of the successes, which I will explain later on. But I did a lot of theoretical and computational research. And then I did my postdoc at QIMR Berghofer in Brisbane. And I was starting from microvasculature, and then I moved a little bit higher now to more network systems neuroscience, but still combining neuroimaging and computational neuroscience in both healthy populations and even across species. And then in 2021, I moved to Monash and did a little bit higher again and more psychology-based brain mapping, structural MRI and stuff. So again, I got my PhD in 2019. So I'm now officially a lot of the grant schemes in Australia, 5 is the kind of the cutoff per se. So I'm now over that. So some people, the definition of ECR is probably flexible across different granting systems. But I wouldn't be able to apply for a DECRA anymore, for example, because that's the cutoff for a DECRA. So 2019, that was my first time to try applying for a grant. Because again, I had a decent number of publications during my PhD, so I thought, okay, maybe I have a fair chance. I have a really cool idea. I think my PhD was cool enough, and I developed a toolbox to apply it to some medical and health related context. Obviously, that was a failure. That I was way off. I was really way off score wise. Well, probably because I didn't know how to write it. I was very young. I had no clue how to write it. But at the same time, I was part of another ideas grant from UQ. I was the CID because I was providing the modeling component of that. And I was lucky, and that was actually successful. So that was pretty good. So that's if that helped me, I think, in the next several years with my application. And then 2019 was the first time that NHMRC did the investigator grant. So towards that end of the year, I tried again. I was just tossing, my luck and see how I can go. But again, that was a big failure. Most of the comments that I had was these guys applying very early on. But I had the EL1 investigator grant was meant to be for people below 5 years. So I was less than 1 year. I thought I could spin it, but I wasn't able to. And then I was also involved with some like little seed grants with collaborators in University of Wollongong. That was successful. So that was 2 mini successes. And then 2020, I really believed in my ideas grant in 2019. So I tried again, but failed. But I was like 0.1 below the cutoff. So I thought that was a really good idea. I wrote it better, but there's just not much money in the country. But at that point, I didn't apply for an investigator grant because I realized, okay, maybe they are right. I'm too young, and I just need to build more with my publications and research track. The results from the seed grant in Wollongong, we submitted that for an AIC discovery project, but that failed as well. So there's been a lot of failures, as you can see. And then 2022, I decided I won't submit anything. I'll just do research and improve my track record. And in 2023, that's when I started building up again. We applied for an equipment grant for an NHMRC within the faculty in Monash, and that was successful. That was across multiple schools that provides money to buy new equipment. So that's good because we were thinking of a new experiment at Monash. I applied for a faculty-based early career fellowship because I needed money to support my salary at that point. And that was successful. And then obviously I applied for an investigator grant, and then that was successful. Hence, I am here as a panelist. And then 2024, I applied for a BBRF. It's still pending. Hopefully that goes through. And we also have an AIC discovery project that got successful in the EOI, and we're waiting for the results of the full application. So you can see over the 5 years, I've tried 11 times, 5 successful, 4 failed, and 2 pending. So I think what worked for me really was I was very diligent in looking at every single grant opportunity. And this is probably something that I will highlight. There are several small internal seed grants within your university or faculty or school or travel grants. They're really helpful to show that they can give you some money. And then I was reading a lot of successful grants from the university banks because I didn't know how to write a grant. So I think that helped me a lot. And I attended every single possible webinar to get as many opinions as possible. And I joined a lot of internal peer review processes where the applicants kind of exchanged their applications and looked at each other's application. Because when you write something for months and months, you're so into it, you actually forget a lot of the details. That's important. And I spent time understanding every single criteria. And that's important too. And then finally, a couple more points, looking back at my experience. I think I tried early on, but I should have not tried early on. Maybe I should have waited a year or two. Just because of the research that I was doing, it was very theoretical and computational, and I don't have many fancy journal publications. And as much as we don't want it to be the currency of the system, it is still the currency of our system. So I had to really make sure I write everything clearly and, provide the message that I want to convey in a really, really concise way. And yeah, networking helped me a lot because I was able to collaborate with many people. At one point, my investigator grant was looked at by 10 people, and I had the comments of like 8 of them in front of me, and I was just looking at everything and trying to see which one's gonna work, and it worked for me. So I think that's it. That's everything that I'm gonna say.
Thanks, James. Quite a whirlwind journey there. Quite a lot of successes. So congratulations for that. I'm going to introduce the next panelist, Dr. Cassandra Wannan. I might be mispronouncing your last name. I apologize.
That's okay. Everyone mispronounces my last name. It's Wannan.
Wannan.
Yes.
Great, thank you. So if you don't mind sharing your screen and taking us through what your research journey has been so far.
I will do that. Just give me 1 second.
Oh, and if everyone just like in the chat, Michelle is dropping like links to each panelist's research website. So if you want to follow up more on the individual researchers here, feel free.
Okay, have you got my actual presentation?
Yes, that's on the screen. Thank you.
Okay, perfect. So yeah, I guess the imposter syndrome is also quite real here. But I guess I have some similar comments to James, and also some practical tips, I guess, things that I've learned over the last couple of years applying for especially investigator grants that may be useful to people when they are putting together their own applications. So I'll go through some of this and I'll try and skip over the bits that sort of reiterate what James said. But yeah, I guess just to start briefly with my sort of research journey today, I guess a lot of people kind of come straight from their undergrad into a PhD and they go from there. I actually worked for a few years in something completely non-academia related. I was working as a statistician at the College of Surgeons for a few years, not really loving what I was doing, and decided I was going to come back and do a PhD. So I came back in 2014, started my PhD, was working on that part-time for most of that. Also had my daughter at some point in among there as well, just to add to the chaos. And my PhD was conferred in 2021. And then I started my first postdoc at the same lab where I'd done my PhD. And I was there for a bit over a year. And then I took up a second postdoc position at Origin, which is where I am now. And as you may be aware, given that I'm a panelist here, I was recently successful in my investigator grant. So I guess I have a similar / maybe slightly more depressing list of grant failures here. So I guess I did something similar. I wasn't really in a super secure postdoc position when I finished my PhD. So I really felt a bit of pressure to start applying for grants. Realistically, I think I would probably also recommend, if you're in a secure position and you have a couple of years funding under your belt, to really focus on maybe some research outputs, at least for the first year or two before you start fully diving into grant applications. But nonetheless, here we are. This is essentially a list of all the things that I applied for following completing my PhD. So starting in 2021, I think I literally just made the cutoff for an ideas grant that for some reason I was CIA on. And that was unsuccessful, and I applied for a whole bunch of other things over the next few years. And out of all of these applications, only 2 were actually successful. This UniMelb ECR grant, which is a small grant, $40,000, and also this Geoffrey Rowe Faulkner bequest, which was another University of Melbourne grant. Again, it was $35,000. So I wasn't really in a position where people around me were putting in grants that I could, be a CID or a CIB or something like that on. So the things that I was putting in, really, I had to lead myself, which probably for some of these larger grant schemes made things a little bit difficult. But in the end, I did get lucky with my 3rd attempt at my investigator grant, which seems to be quite common. So I just thought I would run through a few tips, things that I learned from, seminars along the way, and also from feedback that I'd gotten from people who'd read grants and from reading grants, things that I found really helpful. So the first one, and this is similar to what James said, is that there's a lot of kind of small grants that you can apply for, and getting your first big grant is really hard for most people. And it can be a really nice idea to try and build up a track record in these smaller grants, applying for awards, travel awards, researcher, early career researcher plenary awards, things like that. Things that build up your track record, gives you, small amounts of funding here and there, all these things that you can put on your bigger grant applications to show that the people around you are interested in your work and you are bringing in money, even if it's kind of small amounts of money. And definitely keep on top of the university grants. Some of them are really competitive. I know at Melbourne Uni, there's often one grant for a whole faculty, which makes things really hard. But, it's kind of a numbers game. I think you have to be in it to win it. So if you do have time and you have applications that you can put in for things, it's often worth it. And similar to what James said, is seek out feedback from a lot of people. I wasn't in a position where I was getting direct feedback really on my grants from my primary supervisor, especially in the first couple of years. So I kind of took it upon myself to reach out to other people that I thought would be able to provide me with useful feedback. And to be honest, that has been a real game changer for me. I think definitely with my most recent investigator grant, a couple of the people who I asked for feedback from gave me such pointed and useful feedback that really helped me shape, especially the sort of research impact and leadership sections of my grant. So I definitely think seek feedback far and wide, particularly if you do have university programs where they match you up with a mentor or with someone else who's been successful. At Melbourne Uni, we got matched up with someone who was kind of outside of our area, which also was really helpful for me. I think it can be useful, especially in neuroimaging, to have someone who's not directly in the field give you feedback to make sure it's understandable for someone who doesn't necessarily do neuroimaging, because you never know who your reviewer might be. One thing that I also found really beneficial and something that definitely helped boost my scores was just building up all my leadership activities. So I really kind of said yes to a lot of things, student supervision, which is something that I really enjoy. So that's kind of easy to say yes to. But also things like joining societies and committees in leadership positions. Don't be afraid to put yourself forward for a secretary position or a chair position. These things are really helpful, and not only for grant applications, but also for building up really good networks, getting to know other people in your field. So I would highly recommend that. And also within the university, I've definitely found sitting on sort of committees within my faculty and within my university has been really helpful. Again, not only for my sort of applications, but also for getting good understanding about how things work at the university and, you know, being able to have an impact and a voice for ECRs as well. And also reviewing papers is always good. Something that I found really useful, and I don't know, it'd be interesting, I guess, to get other people's thoughts who are at the sort of EL1 level, but when it comes to kind of describing your research impact, it can be really hard when you've just finished your PhD. You may have, you know, 1 or 2 papers that have some impact. And some advice that I was given was that it's really okay to focus on just one body of work and discuss that in a sort of impact case study. And that's what I did in my most recent investigator grant. And it seems to have worked for me. I got over the line after sort of going from a near miss to a clear miss to a success. And my impact score definitely increased with the last application. So this, I think, could be particularly relevant for people who do more methodological and computational work. If you have a particular paper or a method that has had some level of uptake, being able to describe this really well, you know, who's used your methods, what has it been applied to? Has it been applied to things outside of the specific disorder that you may be working on, for example? You know, who's been writing about it? Has this led to any other big studies in the area? Has it led to any collaborations? So, you know, for me, I had one paper where this sort of thing really fit. And I found that it was really nice to be able to focus on that rather than trying to get these kind of disparate papers that I had into one kind of impact section. And the last thing, and again, I'm kind of echoing James here, is collaborations are fantastic. Don't be afraid to seek out collaborations. If there's someone who's doing something really awesome, or there's something that you're really interested in doing, but you don't know if you have the skill set or the knowledge or you need help, don't be afraid to reach out to potential collaborators. You know, there's a potential it could lead to something really good. And also say yes as much as you can. Obviously, you know, don't overburden yourself, but if people reach out to you for help, definitely try and say yes if you can. I think for me, that's led to a lot of opportunities in terms of publications and, you know, further collaborations and things like that. Another thing, and this is super practical advice, but use headings. So when you're putting together your you know, all your impact and leadership sections, when people are reviewing this, you have to remember that they have a whole lot of grants to review, and making things as easy as you can is usually the way to go. So I saw, you know, I spent a lot of time also looking through the Melbourne Uni sort of grant repository and picked out the ones that I thought kind of flowed the best and had the most level of impact when I was reading through them. And something I noticed was that just having subheadings within each of these categories made a really big difference to how a grant read. So definitely I would do that if I was putting together a grant. In terms of the actual research project, one thing I spent a bit more time on for my most recent application was the first page, and trying to make this as impactful as possible. Again, the advice that was given to me is that, you know, reviewers have a whole lot of grants to review, and you really want to catch their attention in the first page. So I really kind of went with this structure, sort of like an executive summary, I guess, where I kind of laid all the sections of my grant out at the very start. So what's my vision? What is the problem that I'm trying to address? Why is my particular solution the one that you should be funding? What are the broad objectives of my study? And what impact is this going to have? Who am I going to be helping? What sort of change am I going to make? And then I would definitely recommend just putting a nice image on the front page, just to break things up a little bit. So the other thing is, when you're writing these grants, is get really familiar with the score descriptions, descriptors. So, you know, you'll have a marking guide in there. Have a look at what the top scoring grants should have in them, and then use your actual wording from that in your grant. So the investigator grant, for example, there's a section in terms of, you know, the outcomes of the project, and literally you can use those words in your grant. Just make sure you explain why, why you might be having an extremely significant or transformative change or outcome from your project. Don't just put that in there as a sentence. But yeah, definitely, if you can, try and use the wording, make things as easy for the reviewer as possible. Again, something that I found really nice in a couple of other grants that I saw was really clear identification of what the current gaps in the literature were, or the current gaps in the particular study area were, and how they were going to directly approach those gaps. And that was something that I used in my research project. And what I found is I had this in the first year I applied, and then the second year I applied, I had kind of not taken this out, but it was a little bit less clear, I think. And the comments I got from the reviewers really reflected that it wasn't as clear. So in my most recent attempt, I went back to just clearly spelling out what these were, and it seemed to really help. So I literally had these 5 research gaps, and this is just one, and then how I was directly going to be addressing that gap with my particular method that I was using. Yeah, so I guess the next thing, and this can sometimes be hard, but is to focus on translation as much as possible. If you can describe what the real world implications of your work are going to be, I think that's always a benefit. Do you have some pilot data that shows how this might benefit people in the real world? Is there an actual pathway to translation? So even if it is kind of a methods development, how would developing this method at some point down the track lead to some kind of real world implication, or some kind of improvement in the lives of the people that you're focusing on? And the last thing, and again, this is something I saw in another grant that seemed really impactful for me, and I decided to put in my own grant, is a really clear description of who is going to be benefiting from my research and how they'll be benefiting. I think for the first couple of times I put this together, I had a kind of vague statement of what I thought the outcomes would be, but I didn't really clearly spell it out. So for the last time I applied, I had this section, and I said, who's going to be benefiting from this? Clinicians, and how will they be benefiting? Children and young people experiencing mental ill health, same thing, how will they be benefiting? So really clearly spelling that out, again, making things really easy for the reviewer, and, you know, leaving out as much vagueness as possible, I guess. And Sina kind of mentioned to me it would be good if people had any thoughts on, you know, the current funding landscape. I think it's kind of hard. I don't know what else is out there apart from sort of what we're all already applying for, but there is some opportunities in the Wellcome Grant scheme. I don't know how familiar people are with Wellcome Grants at the sort of early career stages, but I know people at Origin, where I am, have had some success with these Wellcome Trust grants, and there's one open at the moment, which I think might be quite applicable to a lot of people in OHBM. So that's worth checking out. And the other thing is that the MRFF scheme is supposed to be, I guess, you know, better funded over the next few years. So some of those schemes that were previously, you know, had very low success rates, like the ECR scheme with its 2% success rate. Yay to anyone else like me who applied for that. Theoretically should be getting more funding pumped into it. So there may be a little bit more money floating around and more sort of a higher success rate, I guess. And that's kind of it from me. But yeah, thank you for inviting me on, and hopefully some of that was useful.
Amazing. Thank you, Cassandra. I think we did have somebody ask in the chat if it was possible to have your slides shared, because I feel like some of those tips were really useful. Feel free to think about whether you want to do that. We can always circulate that after the webinar as well. And I will next introduce Dr. Remika Mito.
Oops. Hello. All right, I'll just share my screen now. Here we go. Can you see my slides?
Yes, perfect.
Beautiful. All right. So yeah, my name's Remika. I'm a research fellow at the University of Melbourne. I am presenting today from Melbourne, so from the unceded lands of the Wurundjeri, Wurrung and Bunurong peoples, and I'd like to acknowledge the traditional custodians of the land from which I'm presenting, and from which I'm really privileged to work and live. And thank you to the organisers for organising this webinar. Yeah, and for inviting me along to share some of my experience. I resonate a lot with what James and Cassandra said about imposter syndrome and being here today, but you know, I'm still at a fairly early stage in my career, but hopefully some of my experiences will be helpful to you. I just have a few short slides today, so I thought I'd start off with a bit of a, you know, short overview of my research experience so far, in case it's a handy benchmark for some of you. So I did my undergrad at the University of Sydney, and I basically really enjoyed neuroscience during my undergrad. Decided that I also really liked research, thanks to a short little research stint that I did during my undergrad. So pursued honours in neuroscience, and I did honours in neuropathology, which, you know, really made it concrete for me that I really liked research, but I didn't really like wet lab work. So that's kind of how I got into neuroimaging. So I moved down to Melbourne to do my PhD. I was working in the diffusion MRI space in Alzheimer's disease. And after my PhD, I was really keen to move overseas for a couple of years for a postdoc position. My partner was finishing off his PhD at the time. So, you know, my plan was that we would move over after a year or so after he'd finished. So I took up a postdoc position at the sister lab from where I did my PhD. But actually that 1 year postdoc ended up turning into a much longer postdoc position, which was, you know, both a really nice opportunity for me because I got to stick around in the same group for a while, but it was obviously a consequence of COVID, I suppose. So I ended up sticking around for a little bit longer. And you know, I suppose it was towards the back end of my postdoc that I really started to think about grant funding. Early on, you know, I really had my sights set on moving overseas. So, you know, I'll talk you through my timeline a little bit in the next slide. But yeah, essentially for the first part of my postdoc, I didn't really apply for any funding. It was only towards the end that I started to apply for some grants. And yeah, I was fortunate enough to get an ARC DECRA that just started this year. So I've moved across to the University of Melbourne, where I'm now starting my research fellow as part of that funding. So I'm still in the diffusion imaging space, mostly working on healthy brain wiring for now. So in terms of, you know, funding and what I've applied for, I thought I'd just talk through this a little bit chronologically. But with firstly with a couple of milestones. So, you know, I completed my PhD mid-2019, so similar to James, sort of at the 5-year scary mark now. But another thing to point out is that I got my permanent residency at the end of 2020. And, you know, prior to that, I was an international student and an international resident. And I suppose this is important for some people because some funding agencies or some funding bodies, you know, their fellowships are only open to people who have residency and only have citizenship. So, you know, for anyone who might be international online, it's something that's important to consider. So it was only after this, and, you know, this is both because I got my PR, but also because I decided that I was going to stick around in Australia instead of going overseas, that I started to apply for funding. So I'll talk you through a few things that I applied for. And I suppose what might be a little bit different to James and Cassandra is that I actually didn't even go for any of those big funding bodies early on. So the first grant I applied for was a really small 20K philanthropic organisation grant, and that was something I found out about through my lab. It was a bit of a touch and go learning experience because I'd never applied for a grant, never really had any research funding. So it was really just an experience in terms of how to manage research funding when what you plan to do you can't really do because of things like COVID. But that gave me a little bit of confidence to start applying for other philanthropic funds. So the next one I applied for was something called a Jack Bruffer Foundation grant. This is, again, a philanthropic organisation. It's got a fairly, niche eligibility criteria. I think it's only open to people who work for a research institute as opposed to a university. And they mostly focus in the pediatric space. And I didn't do any work really prior to that in the pediatric space, which probably played into, the fact that I was unsuccessful for that grant. I then applied for a Brain Foundation grant, again, small 40K research grant from a philanthropic organisation. And, the Brain Foundation is really focused on certain neurological conditions, one of which is concussion, which is a space that I was starting to work in at that time. So I was, fortunate enough to get one of those Brain Foundation grants, which started, giving me a little bit of grant funding or project funding to run my own, independent projects and also get a bit of experience managing, small pockets of money. And it was only towards the back end of 2022 that I really started to think about, okay, I really want to start doing some more independent research and thinking about, fellowships and things like that. So towards the back end of 2022, I was really just applying for quite a few things. I had sort of put aside the dream of going overseas for a postdoc, but I really still wanted to go overseas. So I applied for a VESCI Victoria Fellowship, which is a Victorian state government fund. And that gave me the opportunity to go over to UCL last year for about 4 months. And I also, so I think Cassandra also got one of these, but a University of Melbourne Early Career Research Grant, again, a 40K institutional grant, which gave me the opportunity to do some of my independent research while I was overseas as well. So that was really great. And then it was, the end of 2022 that I applied for the DECRA, which I was, very fortunate to get. But, this is well over a year before you actually get the funding. So you do have to plan for these things, I suppose, with a bit of with a bit of more time ahead. And I also applied for an HMRC investigator grant in that round in EL1, which I was not successful in getting. So I think the last couple of talks have really covered in a lot of detail some practical advice, which has been great. And I think I definitely learned from a lot of that as well. So thank you guys. But what's helped me, I've just got a few dot points. So a lot of this has been covered by the other presenters, but early on when I decided that I wanted to start applying for funding, I started attending a lot of information sessions. There are institutional level ones, there are university level ones. These are really, really handy, and just getting a lot of advice from people who have had success, or the granting officers who really help you with the eligibility and writing up your grant and making sure your paperwork's done in time, that sort of thing, it can be really helpful. And a piece of advice that I got from the info sessions that I thought was really valuable was about knowing your funder. This is particularly important for those philanthropic grants. There's a lot out there. I think often we really focus on those big ARC and HMRC grants, but for me, it was really helpful getting some of those small pockets of funding so I could sort of build up a bit of a profile for grant funding before I applied for a bigger grant. And from that perspective, I think it's really important to know the funder and know what their priorities are and what they really want to be spending their money on. And it goes for the big funders as well. Really understanding the back end of what they want to fund, I think it's really important. This has been touched on by the other speakers, but for me, reading successful grant applications was really, really helpful. Whenever I was applying for a grant, I reached out to people who I knew had been successful and, asked to read their grant applications, and also went on the university libraries to get those successful grant applications to see what worked. And finally, mentors and feedback. I think I've been really fortunate to have fantastic mentors. They might not necessarily be the people you work with. And like others said, you know, sometimes it's good to seek people from outside of your group to get some different advice as well. But for me, yeah, that's been really, really helpful. I don't think I would have had nearly as much success if I hadn't had so much input from others. So I think getting feedback is really, really valuable, and yeah, definitely something that I would prioritize. So that's it from me. I'll stop sharing so next presenter can go.
Great, thank you, Remika. Really
No worries.
lovely advice. All right. And then next person to introduce is Associate Professor Jess Taubert. You're able to share your screen.
Hopefully. Okay, how's that?
Perfect.
Excellent. Okay, I think if I could add one piece of advice to everything that's already been said by all the fantastic talks we've already had, it's that I think grant funding in general is comes a little bit, or you have a little bit more success when you know who you are. I think that's a really important ingredient. You have to know who you are, you have to know what you offer, and you also need to know your method. And we have specific funding challenges in neuroimaging because it's an expensive technique. So I think you have to think about how you sell yourself and how you're going to sell neuroimaging. So on that note, I'm going to briefly introduce me and see if I can sell me and my technique to you. So in general, I'm interested in how we recognize social agents and how we recognize their behavior. And our current model of primate vision assigns visual recognition to the ventral visual pathway and breaks that pathway up into several processing stages. Now, it doesn't matter how this is visualized, you can visualize it any way you like, but the general idea is that when we see an object or a person, that image is projected onto the retina, and that's converted into an electrical signal that is fed into the early visual cortex. That's where low-level visual properties are processed and pooled, and then at the next stage, the incoming input gets compared to stored representations. And at the end of all this, the brain ultimately is able to classify and label the things that we see in our visual environment. Now, the modern metaphor for this hierarchical processing pipeline is the convolutional deep neural network. And these days, our DNNs are extremely powerful and very accurate. It's to the point that you can now feed a completely novel test image to a pre-trained DNN, essentially off the shelf, and it should be able to tell you with high degree of probability that that image contains a dog. Further, depending on the architecture of the DNN you are using, it might also flag the tiled bathroom and the clothing on the floor here. Now, as impressive as our technology is, my argument is that we can read out so much more information from this image than any of our machines. For example, despite not witnessing previous events, we can tell that something unexpected and wholly undesirable has happened here, and instantly we start to infer who is responsible for that disaster. So my conjecture is that the processes underlying our perception of the world go beyond the putative classification of visual features and objects. We see stories and we see the characters in those stories. So me and my lab, we are specifically interested in identifying the neural centers and functions in the primate brain that generate these stories. So my research has been inherently cross-disciplinary, and a lot of that has been kind of baked in via my postdoctoral and pre-doctoral training for that matter. So I was originally trained at the University of Sydney as a psychophysicist in the School of Psychology. So experimental tasks where we measure explicit responses to visual stimuli are kind of the bedrock of my research program. Then I was trained to translate these tasks into comparative experiments to test animal models at Emory University. That was my first postdoctoral experience. And at that stage, I primarily worked with chimpanzees and rhesus macaques, but I also produced some papers using spider monkeys, capuchins, and even chickens. So along the way, I also was trained to analyze and collect eye movement data. This is just an approach which enables a more direct comparison between humans and non-humans without heavy conditioning, so it comes in handy in a lot of circumstances. But turning now to neuroimaging more precisely, I was first trained with fMRI, or in the fMRI technique, by Wim van Duffel at KU Leuven. This is in Belgium, where we relied on functional activity maps to guide single unit recordings in the macaque brain. So this technique, neuroimaging, allowed us to target and compare face-selective neurons in different parts of the macaque brain. And then beginning in about 2014, we also combined fMRI targeting with microstimulation in the scanner to map electrical connectivity within local brain networks. So that was a lot of fun. Then in 2015, I used my functional imaging skills to obtain a senior fellow position at the National Institute of Mental Health in the States. So at that time, my primary goal was to use contrast agent enhanced functional imaging to guide inactivation techniques. So we combined functional imaging with excitotoxic lesions, that's a permanent form of lesion, or Musamore protocols, those are reversible lesions, to effectively shut down particular brain regions and measure the behavioral consequences of that inactivation. However, during this time, I became very interested in measuring task-based activity. And again, still in macaques, and using PPI or psychophysical interactions to estimate task-based connectivity using the scanner. So this allowed for stronger comparisons between human and animal data, and we were able to make really strong inferences about visual and social processing in the primate brain. At this time, I also had a fantastic mentor, Leslie Ungerleider, who really encouraged, a combining and concatenation of techniques. So, it wasn't just about do one thing successfully and build your sort of really monotone methodological progress that way. It was about be diverse, be interested in new things, don't be scared about trying new things, and build bigger collaborative networks in order to do those new fantastic things. So I had a lot of really positive encouragement for building these really large scale neuroimaging projects at that time. So in 2021, I was awarded the ARC Future Fellowship and moved to Brisbane, Australia at the University of Queensland, where I am now. And these days, we use high field fMRI to translate a lot of what I did in macaques during my time at NIMH into basic human research. So it's very likely at this stage I'm preaching to the choir, but it is important to remember that there are a number of very clear benefits of using neuroimaging in certain problem spaces. As someone with a research program that is grounded in non-human primate research, I don't usually highlight the spatial resolution of fMRI because, to be honest, nothing beats single unit recordings. So I put that one aside. And when I'm trying to sell functional imaging, especially to a grant maker, I focus on other benefits of neuroimaging, to include that techniques like fMRI provide a bridge, a very useful bridge between animal and human research. That said, not all animals belong in the scanner, and part of my very first postdoctoral experience was I was meant to train chimpanzees to lie in the MRI bore, and that is a massive cautionary tale if anybody ever wants to talk about that some other time. But yes, do think about what animal you're going to put into the scanner if it's not a human. Not all of them like that environment. But another benefit of functional imaging or MRI work in general is that it's a non-invasive technique. So that allows us to expand our mechanistic questions to both developmental and clinical spaces. And I think that translation has been mentioned by several of the panelists already today. Something that I think about a lot is that scanning sessions can be, while they are expensive, they can be very fruitful, and they produce multiple metrics because we can measure brain structure. We can also measure functional activity, both functional and structural connectivity, and even brain metabolism, all within that one session. So when you're proposing to do fMRI or MRI experiments, it's important to try and capitalize on those experiences to prove cost effectiveness in that environment. And finally, I think doing neuroimaging work in general provides these opportunities for contributing to global scale programs and multi-lab databases, which are popping up all over the place. And these are fantastic sources of information. A lot of people probably rely on these large databases of data to do their work. So I think that's an important point to emphasize when you're talking to grant makers about what you can do with this data beyond its immediate publication and output value. Okay, so I just wanted to quickly touch on the the grant agencies that I've been successful with. If I was to mention all the grant agencies that I've applied for, we'd be here for hours. So I'm only going to touch on the grant agencies that have actually I've had some success with, and that's, you should keep in mind that for every success there is at least 10 failures. So please don't see this as anything to brag about. But my work in Belgium was funded by a fellowship that was via the University of Louvain, which is in Wallonia on the south side of the linguistic border in Belgium. But the actual work was conducted at Leuven, which is in the north of Belgium. And the scanning work in particular was funded by the FWO, which is a grant or a government agency in Flanders that funds a lot of research, basic research in particular. So that's how that work was funded. Then I moved across to NIMH, and I was in the intramural research program, which is distinct from the extramural research program, which funds external agencies. The good thing about the internal research program is that, or the intramural research program, is that we had very large budgets, but the downside was we had to justify them just like any other budget. So there was really blue sky thinking and a lot of collaboration between very large groups, but the experiments had to be, really as good as they can be. There were no excuses, let's say. And the skill sets that we acquired under those in that environment were quite immense. So the training was very rigorous, but what we missed out on potentially was we weren't allowed to apply for external funding. So I stayed in contact with or sharpened my grant writing skills only through contact with the extramural program and trying to understand what was fundable in that space. But nonetheless, moving back to Australia, I still maintain some funding from the NIMH, so I still do a lot of work externally on that campus. But the ARC Future Fellowship is obviously funded by the ARC. There is some research funding involved in that particular program. I also have a project that's funded by the BBRF, which has been mentioned already by a few others. And I also have some internal funding from the University of Queensland. This has been mentioned by all the other speakers as well. So our internal sources of funding are really important. And I wanted to specifically point to the Centre for Advanced Imaging at UQ, because that's one of the mechanisms in which I can collect pilot data in the scanner before I actually go to grant makers and propose bigger, more expensive experiments. So I suppose that was where I wanted to end it, and I'll just sort of advocate broadly for not only collaboration. I think that's been mentioned as a point for getting a really good track record of grants going, but I also still continue to apply to international government agencies for grant funding. So at the moment, I currently have grants submitted to the NIH through the extramural program as an international contributor, also the FWO, again, as an external person. I think the Human Frontiers of Science program also has a really great program, particularly for early career researchers. And again, these are all international bodies and, both private and government grant makers. And I think, while you're doing your grant research, it's also worth expanding that lens and looking at what other international bodies are funding as well. So thank you. I'll try to stop sharing this.
Thank you, Jess.
No problem.
Quite an interesting research trajectory there. And lastly, I'm going to introduce Associate Professor Max Shine.
Hey everyone. All right. Last little presentation before we get into the Q&A. So I was racking my brain a little bit and thinking about what to talk about. And we've already heard from 4 really talented young researchers that I think have covered a lot of the nuts and bolts, the nitty gritty, the kind of different opportunities. It's always very difficult going last in one of these types of presentations because a lot of what you need to know has already been said. So I thought I'd take a slightly different tack and change the title from Tips to Success to something different, which is the Wu-Tang principle of grant funding, which is that cash rules everything around me. Get the money, dollar bill, y'all. So we don't talk about this near enough because it's really depressing. But no matter what kind of scientist you are, it doesn't matter if you're electrophysiologist, a theoretical mathematician, someone who looks at slices, someone who does neurochemistry, someone who just, talks about being a scientist more than actually a scientist like myself. The Wu-Tang principle applies to all of us, which is that the money for research is just really, really hard to come by. And, it's really difficult world to live in because you have to essentially be proving yourself, I think, every single moment of every single day. In fact, I'd say there are 3 perfectly acceptable responses to this this strategy or this world that we live in as modern researchers. One is to run away as fast as you can. In the US, which is an area I know a little bit just from collaborators, there's been a mass exodus actually of young, talented PhD students into quite lucrative industry careers. In fact, a lot of Australians have done it too. And I think we need to, as a community, acknowledge that this is actually a really great set of outcomes and opportunities for young scientists. There's heaps of different places you can go. There's companies that explicitly use neuroimaging, but don't forget, anytime that you achieve a PhD, you're essentially showing a potential employer that you can put up with an absolute mountain of nonsense and BS for years, tons of poorly designed projects, multiple different parties, and you can work through to completion and finish studies. These kinds of skills are actually really applicable across the business world. And so don't doubt yourself as a young scientist if you're not eager to kind of compete for the crazy small amounts of resources. This is a perfectly acceptable response. Another one is heavy drinking and screaming into the void. I typically like to do this with other academics so that we can talk to each other about how difficult this is, discuss ideas, kind of like we're doing today, reach out to people. Everyone's been through similar kinds of experiences. For every one success you have in academia, there's at least 5 to 10 absolutely gut-wrenching failures. And everyone's got these. The imposter syndrome is absolutely real all the way through. And I think one thing that kills imposter syndrome is taking a growth mindset. And the second thing is surrounding yourself with colleagues that you can bitch and moan to, that understand how hard it is. So I'd really strongly encourage you to do that. But the third response, and what I'll talk a little bit about today, is really owning it. So if you have to live in this world of scant funding, what can you do to kind of maximize your success? You've already heard a lot about it today, but I'll keep it more broad in my talk. I like to use the KISS principle in my group, which is just to rock. Actually, no, it means to keep it simple, stupid. I think a lot of the time, particularly with neuroimaging and neuroscience, we forget how just mind-numbingly complicated a lot of the different logics that we use to understand the system in front of us are. We use techniques that are really, really complicated to people outside of our field, but yet we kind of are a bit ho-hum about it. Oh, it's just a really, really simple linear algebra approach or something. Most people that work in science don't deal with maths on the level that we deal with, at least the kinds of people that are going to be assessing your grant. So try to keep things really simple and understand them really well before you proceed, and do a little bit less of the kind of pushing on how clever we are. I think we need to be a bit better about explaining what we do. In terms of the KISS principle, this is my first little GIF here. This very talented juggler can obviously do incredibly difficult things, but I think oftentimes it's really not that impressive to people if you say that I can do some really, really complicated thing like juggling all these balls and landing on your chest, when to them they can't even juggle 2 balls, let alone 3, let alone 7, or what she's got. So be really, really careful when you're kind of talking about your techniques. Make sure that they're interpretable and applicable to people that aren't in our field. A second thing I think which is really, really underrated is to be able to point directly at the problem that you're trying to attack. Find the gap in the literature, find the problem that you can see, and show exactly what it is. And when I'm reading grants, particularly neuroimaging grants, a lot of things will be things like, oh, we don't really understand this, and then we're just going to do this technique. I don't think that's enough. We need to really point to what the exact problem is, what the hypothesis-driven science is that's going to take us out of that problem, to reduce our uncertainty of that problem. And I'd really encourage young scientists to really think carefully about identifying their problem with clarity. As I mentioned before, I think a collaborative approach is really, really important. You've heard from multiple of the speakers today that working in a collaborative group is deeply important. I think that's now isn't true now more than ever, particularly for neuroscientists. We simply can't be experts in all of the different techniques and imaging modalities that we need to answer the really pressing problems in our field. So you've got to work with clinicians, work with neurophysiologists, work with cognitive scientists, work with physicists to get really good signal. And I think the grants that I see that I'm most impressed by are the ones that take that multidisciplinarity really seriously. And I think as well, kind of wrapping around all of these things is oftentimes we need to find simple solutions to problems, and we find these really complicated ones and we think, oh, I could just do this next. But ask yourself, is this really the simplest way I could solve the problem? Now, just to cover some strategies for standing out when you're writing your grants, I think there are kind of two main strategies. You can be either a positive person or a negative person. In the positive category, you can find a really good idea and say, I think that this is a big problem. This is a big question. You could point to something that you think is really exciting, something new, something really beautiful, something really what you think is important. Or you could do a little brag and say, of all the people that could do something like this, I'm the best. Now, you could also be negative in each of these cases. You could say that I think that the person that had an idea over there had a really bad idea, and you could try to point to exactly what they did wrong with their idea. You can say that you think that something that someone else thinks is nice is actually not very nice, like a particular technique isn't very nice, or a modality isn't very nice. Or you can go for an ad hominem attack. Now, I do not advocate for this one. And to a lesser extent, I think this one's also kind of crappy. I think if we focus ourselves on the problems themselves, the techniques, the questions, and we try to focus on being constructive, we can take both the positive and the negative approach. You can say, I think this is a really cool problem. This is a really cool technique, or I think this technique is flawed and I'm going to test exactly why and show you exactly why. These kinds of strategies can be really good for creating a really constructive community around neuroimaging. Just to put things in a bit of context, but I'll whip through this just because so we can get to the Q&A. The thing that I really wanted to underscore, rather than anything about funding in particular, is just this notion of getting funding from lots of different areas. So I got my PhD back in 2013. During my PhD, I had a small little fellowship from the Rotary Foundation, so that was philanthropic. I then worked on some Michael J. Fox Foundation funding that I didn't myself procure, but I was the beneficiary of. I then was awarded a CJ Martin from the NHMRC. That took me overseas to the States. When I came back, I got a small bit of funding from the University of Sydney, which then translated into a larger fellowship called the Robinson Fellowship, which is now defunct. They now have something called the Horizon Fellowship, but it's really an internal funding opportunity. I then translated that into an NHMRC EL2 fellowship, was awarded a philanthropy fellowship, and now just got an L1 to give me that kind of leadership capacity at that broader scale. But the thing I wanted to point out here is that these are all from really different places, right? You've got philanthropy leading into government funding, leading into local funding, back into government funding, philanthropy. So there's a lot of diversity in these types of funding approaches. And I've also gotten some funding collaboratively with other folks from the ARC and the NHMRC, and then I'm part of what's called a synergy grant, which is a little different. This is multiple different CIs working together, and these are great for doing collaborative science, but they're not really great for supporting research programs for yourself. They're really good for being a part of a bigger, broader community, but the amount of funding when you take these large grants and you chop them up into smaller pieces really aren't enough to kind of support you and your team. So you've got to be really, really judicious about how you choose to bring these things together, particularly because grants now from the NHMRC and ARC, you can only have 2 of them active at the same time. And so now, because of the Synergy grant and my L1, I essentially can't apply for an ideas grant for a few years. And the good news is that newer fellowships have quite a bit of extra money for employing staff and for being able to conduct your science. So that's great, but it's really important for you to be thinking about how to diversify across these different areas. So this brings it back to the Wu-Tang principle. You need to diversify your bonds and to really think about spreading out your funding across these different areas to support you and your group. Probably the main thing that I think is important about funding for young scientists is to really take the mindset that you're essentially trying to buy your way into financial independence. You're trying to demonstrate that you can conduct the research that you see as really important so that you can do the things that you think are cool. Whether you think skateboarding, in a back alley with a cool sweater on is cool, or eating spaghetti out of a bag, it doesn't matter what it is. As long as I think being able to provide you with that capacity, that autonomy to do the science you want to do is what's going to really advance our field and continue to make inroads. Along these lines, I would really encourage you to put in the grants that you care about. When you read grants from people that are putting in a grant that they see as an opportunity, but maybe one that they don't really care about, I feel like it somehow comes off the page. Enthusiasm is so important when it comes to these kinds of pitches, particularly the front page. If you really care about this, if you can really put your finger on what the problem is and say why you as a individual or you as a consortium or a larger collaborative group are the ones to solve that problem, I'm super excited and I want to help you achieve that vision. And I think as a general rule, scientists are really supportive of one another if we can get behind why we're doing it rather than just what we're doing. So you can think of a few different categories here, but you want to bring something in that you think's beautiful. You want to remove an obstacle from the way of stopping people understanding a particular problem, or you just really, really want something really, really bad. Bring that out in your front page. I want to say as my final point, I think sometimes we do a really bad job of supporting each other. And I was talking to a senior scientist, a neuroscientist, a couple of weeks ago, and he was actually telling me that neuroscientists actually judge each other's grants way, way more harshly than other fields like in cardiology and hepatology and some of the other sciences. I think we view this sort of competitive, I'm going to climb to the top of the hill first kind of a mindset to our detriment as a field. And so I think we should think very carefully about that and think about how we can support one another to do our best science by being constructive. And at the end of the day, let's not forget we're a community. We may all dance differently, but hopefully we dance all together as a coherent whole. And with that, I'll move on to the Q&A.
Great, thank you, Mac. Nice analogy with the Wu-Tang. There was a reason why I called Mac last. To get hopefully people more actively engaged in now our Q&A session for the panelists. So I can see someone's already asked a question. So many thanks to the panel for such a great session. Do you have any tips for building collaborations? Is cold emailing ever okay? How do you establish new networks beyond your own lab? Tips for dealing with supervisors / institution policies. So I'll open that up to whoever's keen to kind of give an answer to that one.
The simple, most straightforward answer is cold emails are great. I always read them, and there is some filter where you're looking for, AI generated emails or something, but you generally do read them. And if it's an exciting idea, I would say that I would lead with the science and not with, your CV details, because we're all scientists and we like science. So I think cold emails are a great place to start when you don't have, say, opportunities to go to conferences or other networking opportunities.
Thanks, Jess. Yeah, I like my own experience. Cold emails have led to some lovely collaborations, especially when you just reach out and say to someone I really enjoyed your work, or point out one particular paper that you might have read that's got you really excited.
We're
Often,
very susceptible to flattery. Yeah.
Yeah, exactly.
As a community.
Anyone else have any tips on building the collaborations?
I would say with cold emailing, be succinct if you can. I think when I've had success in the past, a lot of people that you want to communicate with are really busy. And if you send them a big, long, opus of why you want to work with them, they're like, ah. But if you just say really quick, hey, love your work, we do this kind of thing, would be really cool to meet up, or can I send you some more information? I think that's a really helpful strategy.
Great. Let's move on to the next question, just because we're running out of time. Have you found funding specifically targeted to your research area or career stage to be valuable? For example, funding to a study of a specific disease or to support ECRs with research with family, or specific funding for gender-related inequalities?
I've heard actually good stories about people who study very specific diseases, that the success rate is really, really high. And there's a lot of philanthropic foundations who actually focus on very specific diseases. You just need to find them. And that's the tricky bit. So you always need to go to your university website, and they always provide all the details of all these things that you have actually never heard of. Because ARC and NHMRCs are not the only grant institutions. There's so many of them. So I think it's very valuable to find those targeted ones.
There's a lot of yeah, I was gonna similarly say there's a lot of like neuro-specific conditions where there are quite a lot of funding bodies. So I think, for example, like Fight MND, if you're working in the ALS space, there's, Dementia Australia, Dementia Research Council. There's a lot of yeah, disease-specific ones that can be really valuable, I think.
Great. I just put, to keep sort of more questions available to the team, I just put together a little like panel vote on a selection of questions to know which one we should lead with for the panel. And it's looking like the first one. So what's been the most helpful solution to your funding challenges? Or, oh, that whole question is not, I can't even read it. What's been the most helpful solution to your funding challenges, or are there any choices you wish you had made differently, like in summary, through your research journey so far? That's a hard question.
I can reiterate real quick, Tash. I think you can just tell when you read lots of these things, when people have thought long and hard about a grant, they've spent time on it, they've spoken to lots of people, heaps of people have read it. Just the choice of words, don't try to do it last minute, really think carefully about it. And yeah, just take it seriously, long term, plan ahead.
Yeah, and I also got advice at some point that don't just apply for the sake of it, I think. But obviously we all fall into the trap, right? Because we need to survive for independence. But you can tell actually people who've just decided 2 weeks prior to the deadline that, all right, I'm gonna submit one. It shows.
Oh, we've got a question in the Q&A. No, but I think that's like really good advice. That's probably like the biggest one I've taken away so far is like, as an ECR, do not just like feel like you need to start applying for big bodies of funding. It's probably better to try and go for smaller things that you're more eligible to get, and then focus more on your own like research and building up your publications, especially when you've just finished your PhD. And so the question is everyone aware of the Australian National Imaging Facility, which can help people get started, get pilot data, as well as improve your research proposals? So I'm not sure if any of the panelists have used this before. So this is sort of how they have like 14 satellite locations across Australia, with subsidized funding for conducting different sort of neuroimaging from PET scanning, CT scanning, MRI, with also experts in the team that can sort of help with the analysis and collection of the data. But I think the only thing to note is like they don't have their own specific funding, but they can support the neuroimaging community with their research, which is good. And then I've got a couple more questions here. If no one else has any more open sort of questions in the chat. What do you think of collaborative grants? And what factors do you consider when you're deciding whether to join collaborative funded projects? And the reason why I ask that is because now there's been sort of that change in the number of active grants that you can have at the same time. It sort of makes you probably want to be a little bit more considerate in the different grants that you want to put your name down onto and be involved in.
I think early on in my career, I really thought that getting grants was this kind of means to an end. Like if you got a grant, you could put it on your CV and then you could kind of like pat yourself on the back and go, okay, done. But I think there's been a real sort of mindset shift for me that's been very helpful, which is you got to really think, why do I need a grant? What kind of work could I not do if I didn't have the grant? And what could I do if I got it? And if you think that way, then it's really important to sort of apply for grants or put yourselves into situations where the grant will reward you. That's why I wanted to bring up the idea of a synergy grant, right? This is fantastic. And I love the kinds of scientific questions that can be answered by a group of 10 different PIs. But it's not really that helpful if you're worried about supporting a postdoc salary, because the best you're going to do is maybe a quarter to a third of a salary or something like that. And that's, you know, kind of a lot of pressure to put on you as a young PI if you're thinking, man, I've only got 2 slots in NHMRC. Do I go up for one of these bigger grants or do I go for a smaller one? So you got to really think carefully about what you're getting out of it. And I would really encourage you, all the ECRs on the call, to be not really sort of shortchanging yourself. Don't just go into a grant and think, oh, I've got to fight to get on as a CIG or something like that. Think, what do I need out of this? What can I do? What can I bring to the table? And what do I need out of it? And I think a grant that you don't get to go on early is actually probably better for you if you're not getting the funding. Maybe people will disagree with me on this one, but I think you really need to be thinking judiciously about, what do I get out of this, and how is it going to help me enact my research program, and build up those smaller grants first before you go for the big ones.
Following on from that, I think it's just really important to know who you are in terms of where you're at, in terms of what you do well, and in terms of where your research is going. So a lot of introspection and strategy is involved in joining those bigger collaborations.
Yeah, I was going to say, I guess as an early career researcher, it's something that it kind of plays on your mind a little bit, because we can get investigator grants, which are awesome. They give us a salary for 5 years, but then we don't have the project funding. So we either need to try and find project funding somewhere else, or we need to have a data source that we can work on. So yeah, I think it's really hard to toss up what the best approach is, but I think having independent salary is such a bonus at an early career stage that, yeah, it's hard to kind of say no to something like an investigator grant, I guess.
Yeah, that's sort of useful advice.
I might also add that, sorry, just on top of what Cassandra said as well, that, you know, sometimes these philanthropic grants are really good for ECR funding project grants. And so, you know, you're not blocked out of those when you're on an NHMRC or ARC grant. You're still eligible for them. And, you know, that they might not be nearly as much money, but 40K is nothing to, you know, nose up to. So, yeah.
And then I
The nice
think
thing about those grants is that they will often have people who will talk to you very specifically about what they find tractable and fundable. So they're often very precise in their synopsis. So that can be nice. You know, the ARC and the NHMRC, there's a little bit of magic involved there sometimes, but you know, the private grant makers will often be easier to deal with in that way.
And we've got one sort of, I guess, like question / bit of advice from Sid. So maybe Jess, you can confirm this, but I've heard internationals like Australians are eligible to apply for NIH funding, even without American collaborators. Do you know?
It's true. I can confirm.
There you go. Well, that's
Not
exciting.
NSF.
I didn't know that.
So NSF grants are for American citizens. And I'm actually a US permanent resident, but I'm not eligible for that either. So it's only for citizens who are in an American institute. But the NIH, you can even be they say CI or they say PI, I forget, but either way, it's the opposite of whatever's in Australia. But you can be the chief investigator and lead an NIH grant.
You just have to be able to justify that the work that you do, or your capacity to do it, is requires it to be done offshore. So if you were like trying to say that you do the world's best object genetics of mice, they'd say, well, why aren't you at the Allen Institute then? And you have to then justify why your kit is better than someone else's or something. And so for human neuroimaging, it's not too tricky, because the kinds of methods you might do might be different, or the kinds of group you might have might be unique. But yeah, it's really just about justifying it.
Hello. Maybe you do neuroimaging on kangaroos or something. For instance.
Exactly. Or like platypi. Platypi are way more interesting than kangaroos. Why don't we have them on our flag, right?
Because
Agreed.
they're
Let's
so
do
hard
that.
to find. I still haven't seen one.
What?
Lived my whole life in Australia and I've not seen a platypus.
Oh dear.
I know, very disappointing. Does anyone else in the Q&A have any sort of other questions? I think like I have one, but I'm trying to think of the way to word it. So a lot of the grants have components in it about sort of like addressing a specific disease. But what if your research in general, or what you're interested in, is more sort of just trying to understand the brain, and then from there, if you understand the brain better, you're more able to understand the sort of pathologies that affect the brain. I think sometimes like it can be a struggle to know whether you should just try and write a grant that has this more specific focus to one disease, or write the grant on like the general understanding of the brain and more of like a basic science question. Is something that I have struggled with trying to get my head around.
I suppose, I suppose one thing to note there is the different funders, right? So I suppose if you're going for a bit more of a basic science question, then, you know, you're probably not well placed to go for an NHMRC grant, maybe. But ARC will welcome you with open arms. So, you know, it might just be about finding out, I don't know what others have to say, but I think, yeah, just understanding what funders are going to be best for the research question that you have in mind is really important.
I could probably add, because I'm a methods person, right? So I can apply my methods anywhere. My EL1 is actually applying it on like 8 diseases. So, but I just need to write it really, really clearly, because it may sound like a hodgepodge for someone because you're applying it to 8 diseases, but you have to tie them together. That's the difficulty. And I had some help with, within the university on how to finesse that, and it worked. So it's possible.
Well, if we don't have sort of any last questions that anyone wants to ask, or if the panelists have any sort of last bits of advice, I think we might sort of wrap up the session. I'd just really like to thank all the panelists today. It's been really, really useful. Surprisingly, I feel more hopeful, which doesn't make, I don't know. I feel like now I have tips, if that makes any sense. Whereas I actually started the webinar aware that I would be probably feeling quite anxious up. So thank you all for such good sort of encouraging advice. And if anyone has sort of any other questions, feel free to reach out to OHBM Australia. We've got a new website. So we'll be uploading recordings of the latest webinar on the website as well.
Tash, can I just say, because I know the investigative grant is due in a month or so, I'm happy to read and help if anyone is submitting. Just send me an email.
Great. Thank you so much, James. It's very,
I agree.
generous.
Send them all to James. Yeah.
No, send it to everyone.
Throw everyone under the bus. All right. Thank you, everybody.